Good afternoon, everybody. My name is Abby Bronson, and I'm the SVP of Research Strategy at PPMD. We're honored today to have Wave Life Sciences here to give us an update on their stereopure exon skipping program they've been working on. They've been doing wonderful work for our Duchenne community, and so I really want to thank them for taking the time to speak with us today. Before I introduce our speakers, I want to let you know, as I always do, all this housekeeping, I want to let you know that we will have time for questions at the end of the talk. So if you have a question or you want something explained, you can submit your question in the chat box, and at the end of the speaking portion, we'll answer as many questions as we have time for, and those that we don't have time to answer, we'll answer by email. So this will all be posted. We're recording this webinar, and the webinar plus any questions that we get answered, we'll post it at a later date. So now it's my pleasure to introduce our speakers for today. First, we have Michael Panzera, who is MD, MPH, and he's the Chief Medical Officer at Wave Life Sciences. He oversees Wave's portfolio of therapeutic candidates. He's got 17 years of biopharmaceutical industry experience, working at companies Biogen and Sanofi Genzyme, developing therapies for neurological disorders, so lots of great experience. We also have someone who I think many of you may know, Jeff Smith, who's the Director of Patient Advocacy at Wave. He has over 18 years in the biopharmaceutical industry, so I don't know who amongst them has the most experience, but they're both really experienced, great people to be working in our space, and we're happy to have them here. So I am going to turn it over to Wave now, and I think that, Jeff, you're the person that's speaking first.
Yes, thanks so much, Abby. It's our privilege to be here. Thanks, Kaylin, for helping to coordinate the opportunity, and thanks to everyone for being on today. I'm Jeff, and I'm excited to be here. I'm here with Mike, and we want to not just share our work and our research in Duchenne, but also hear from you. So I think it's a really valuable opportunity for us to be here. So get on there, get in the chat. We want to hear your questions, your comments, and make this more of a conversation. So let me start with our forward-looking statement slide. And, this is a slide that may be familiar. It's probably familiar to many of you. And I think for me, it's just a good opportunity for us to pause and reflect on the fact that, we are a science and a research-based organization. And with, inherent in science and research is uncertainty. And so we use words like this, like may, will, should, expect, plan, potential, and hope. And, that kind of reflects the uncertainty there with a science-based organization. That being said, the other side of that coin is hope. And we're obviously very hopeful for our research and the research of our peer organizations in the community. It's just, it's going to take a lot of hard work and continued collaboration from us and from all of us in the community to realize that hope. So that, in a nutshell, is what this slide means to me. So without all the words. So let me tell you a little bit about Wave. We are a genetic medicines company working on delivering transformational therapies for folks in living with Duchenne and other genetically defined conditions. And we do that with a focus on stereopure oligonucleotides, and Mike will talk much more about what we mean by that, but essentially oligonucleotides, as many of you are familiar, are short stretches of DNA or RNA, and they can be used for things like exon skipping to kind of work on gene expression. And the stereopure aspect, so what we're working on there is making controlling the positioning in certain aspects of the investigational medicine in order to create a single defined structure, which we hope will improve the properties in the body. And so, that's our focus. That's who we are and what we do, what makes us unique. And that platform has been applied across a pipeline in Duchenne, including Suvodirsen, which is in a phase 2/3 trial we just initiated in boys that are amenable to exon 51 skipping. We're going to talk a lot about that today. And we have additional research behind that against additional exons. So let me now, Mike will talk more about that. In terms of the company, so I know, many of you aren't quite familiar with WAVE, and that's completely fine. That's all right, and that's understandable. I think there's a reason for that, and the reason is reflected on this history timeline. So we have been around since for about 10 years, since 2009. But if you kind of focus on the bottom half of this slide, kind of the clinical milestones, we've really been focused on, as quickly and urgently as possible advancing our research. And so you see, soon after coming together as WAVE back in 2013, we've been able to, here in 2017 and 2018, advance into the clinic with our Duchenne research, in addition to the other therapeutic areas that we're working on. And so shifting over to the top, a couple of corporate milestones that I'll highlight here. First, we have 2 collaborations with large pharmaceutical companies, and those aren't necessarily in Duchenne, but I think it reflects the fact that folks have been willing to collaborate and partner and support our work, and we're learning a ton from each of those collaborations, and our learnings are able to be incorporated into our research across the board, including in Duchenne. And then also I'll just highlight, our manufacturing facility opened in late 2017, and that was, a purposeful investment. We recognize the importance of manufacturing and supply, and so, we purposefully made that investment to open our own manufacturing facility back in 2017, and that's been important for the company. And so finally here for me today, you'll hear a lot from Mike about, again, what makes our science unique, but I think what also makes us unique is our culture and the commitment to listening, to learning. We're a listen-first organization. We're inspired by your stories as families, and we aspire to help you as much as we can. That's why opportunities like this are critically important for us. Mike, again, will share a little bit later some specific examples of where we have heard, we have learned and listened to the community, and we've changed how we've conducted, where we've conducted our research. And so, it's critically important for us. Everything we do is around addressing the needs and the priorities of the community. So thank you for all the folks on the line, all of you that have supported us. PPMD has been a great partner, as have the other organizations, and we look forward to continuing to work with you to advance our research. Thank you, and I'll turn it over to Mike.
Thanks, Jeff, and thanks to everyone for joining today. It's really a great pleasure to be here to discuss WAVE's approach to DMD, and also to share a bit about the Distance 51 Phase 2/3 program, which is recruiting as we speak. So to talk a little bit about what WAVE does, our approach involves development of therapeutic oligonucleotides. That's what Jeff alluded to. And where we are now is really following 25 years of innovation in the world of oligonucleotide therapeutics. The techniques that we have now are really advanced, where we can precisely control the characteristics of these molecules, and this is what WAVE was founded on, and this is what we call PRISM. The concept is that by precisely controlling the chemistry of oligonucleotides, we can potentially create oligonucleotides that we call stereopure, with improved characteristics as compared with oligonucleotides made by the traditional way, which is known as stereorandom. It was the observation back in 2016 that by controlling stereochemistry, we could improve exon skipping in a dish of cells taken from the muscles of boys with deletions amenable to exon 51 skipping, that led us to embark on the Suvodirsen program and the other exon skipping programs that we are currently doing. Now, to understand a bit about exon skipping, we have to talk a little bit about the background. First of all, it begins with dystrophin, and many of you know this, but so I'll try to be brief. But dystrophin is the largest known gene in the human genome, about 2.2 megabases, composed of 79 exons. So the idea here is that this is a very important protein, and I think its size speaks to that. It's responsible for maintaining the integrity of the muscle cell. So when you lose dystrophin, when it's absent, the muscle cell is unable to function normally, and it eventually breaks down. And this leads to those symptoms that you all know so well. So what we try to do with exon skipping is to allow the body's natural mechanisms to facilitate the widespread restoration of functional dystrophin. And I'm going to talk a little bit about how we're going to do that in the next few slides. So to understand a bit about skipping, one, again, starts with the gene. This is a schematic of the full-length dystrophin gene transcript. So this is what the body essentially reads and turns into a dystrophin protein. So this is what a normal form would look like. Now, what happens in the case of a deletion, you get this gap. You find this place where the transcript becomes essentially unreadable. And what that results in is no dystrophin protein being produced. So the concept of exon skipping is to try and rejoin that transcript, bridge that gap that has formed because of the deletion, to allow the gene to essentially be transcribed and translated into normal dystrophin protein by targeting a certain exon and then allowing the remaining exons to join together. And the thought is that by doing this, you restore a functional protein that then can lead to the restoration of muscle strength. So why do we believe stereopure oligonucleotides are a good approach to treating DMD? Well, one of the advantages of stereopure oligonucleotides is shown here, namely the ability of stereopure oligonucleotide to get into the nucleus of the cell, which is where skipping occurs. This is an experiment where we took cells derived from patients. We put these cells into a petri dish and treated the cells with either a stereopure exon 51 skipping molecule, like Suvodirsen, or a stereorandom molecule that targets the same place in the gene. We then looked at the cells under the microscope and counted the red dots, which indicate the location of oligonucleotide in the cells, and that's shown here on the right-hand side. You can see those red dots in the blue, which is the nucleus. What you can see is there are a lot of red dots in the nucleus in the cells treated with stereopure oligonucleotide versus stereorandom oligonucleotide. Now, what this means is that the oligonucleotides are better getting into the nucleus. So that leads to the question, why is this important? And we believe it's important because the more oligo that gets into the nucleus, the more likely it is that skipping is going to occur. And that's what is illustrated here. This is a Western blot. This is the way we measure the levels of protein produced. In this case, we're looking at dystrophin protein, both full-length and normal muscle, as shown all the way over to the right, and a slightly shortened version produced by those same types of cells in a dish. These cells were taken from a boy with a deletion from exon 40 to 50. After treatment with Suvodirsen or other oligonucleotides, including a stereorandom, what's called a stereorandom TS oligonucleotide, or a stereorandom PMO oligonucleotide. The green bars near the top of the gel are the dystrophin protein. Brighter bars mean more protein. We then compared the amount of protein in these lanes to the level of protein in a normal muscle, and that's what you're seeing all the way over on the right-hand side. The Suvodirsen lane has the white box around it. What this shows is that Suvodirsen and these other two-wave stereopure oligonucleotides produce very high levels of dystrophin relative to those stereorandom oligonucleotides. The numbers here represent the percent of protein versus what you would expect in normal muscle. So in other words, Suvodirsen-treated cells produce dystrophin at a level of 52% of what would be seen in normal muscle as compared to these stereorandom oligos. Once again, this made us very excited, and it was really the first indication that Suvodirsen had the possibility of leading to higher levels of skipping and dystrophin expression than had been seen to date. And this led us to move forward with the Suvodirsen development program. So this slide shows an overview of the program. As you can see, it's pretty comprehensive, consisting of a multi-center global phase 1 study that was just completed on the left-hand side. There's an open-label extension study for patients who participated in the phase 1 study, where we expect to have data from muscle biopsies in the later part of the fourth quarter of this year. And on the right-hand side, you see DYSTANCE 51. This is a global, multi-center phase 2/3 study to evaluate the efficacy and safety of Suvodirsen, namely its ability to both restore dystrophin production and improve function. This study is currently underway, and I'll describe the details of that a little bit later. But I'll discuss briefly each one of these phases in the coming slides. Beginning with the phase 1 data, we found that, as we presented previously, that Suvodirsen was generally safe and well tolerated at doses up to 5 milligrams per kilogram. The most common adverse events that we saw were those associated with infusions. That's typically happening within 24 hours, mild to moderate in intensity, and resolving with symptomatic treatment. Fever, headache, vomiting, and a rapid heart rate were some of these symptoms. And what we saw is that similar symptoms occurred with increased severity at doses above 5 milligrams per kilogram. While this slide illustrates what we saw after a single dose of Suvodirsen, we were obviously interested in what would happen once multiple doses were given, particularly at the doses to be administered in the phase 3 study. And these results are shown on the next slide. So this slide shows infusion-associated symptoms that occurred with continued dosing, and they appeared to decrease over time. Let me walk you through that. So once patients were finished in the phase 1 study, they were eligible to receive weekly Suvodirsen in this open-label extension study, or OLE. This is a graph of the proportion of patients with any symptom within 24 hours after the start of infusion. And this is the data as of the most recent data cut on June 18th. What you're seeing here is proportion with any symptoms on the vertical axis. So that's the proportion of patients on the vertical axis. While on the horizontal axis, you're seeing the number of doses given and the number of patients who received that number of doses. So what is clear from the graph is that it appears to be a decline in infusion-associated symptoms with continued dosing. We've also had no study discontinuations to date in patients receiving these 2 doses. And every week since then, and ongoing as we speak, the number of patients continues to grow. So we will wait to see if this pattern continues to hold. And we eagerly await for these data as well as the dystrophin measurements for muscle biopsies, as I mentioned, in the later part of the fourth quarter of this year. So for the last part of my presentation, I'd like to talk about DYSTANCE 51, which is our phase 2/3 study that is currently recruiting. This is a phase 2/3 study that I have to emphasize was designed with input from the entire community, from investigators, from regulatory authorities, but most importantly, you, the patient community. It's a global study. We're anticipating enrollment in the U.S., Canada, Europe, Australia, Japan. We're targeting approximately 150 to recruit 150 boys with genetically confirmed diagnosis of DMD with mutations amenable to exon 51 skipping. Patients will receive weekly intravenous doses of Suvodirsen or placebo for 48 weeks, and then after that period, all patients will be able to receive open-label treatment with Suvodirsen for another 48-week open-label portion of the study. So all patients will receive drug, and that's what you're seeing here on this slide, which shows the details of the design. When patients come into the study, they'll be randomized to receive one of 2 dose levels of Suvodirsen or placebo for that 48-week double-blind period. Then patients will be eligible to receive Suvodirsen during the 48-week open-label phase of the study. All patients will have a baseline biopsy and then a follow-up biopsy at one time point, so either 12 weeks, 22 weeks, or 46 weeks after starting treatment. This design allows us to collect critical information about Suvodirsen's effect over time while eliminating a need for any patient to undergo more than 2 biopsies. The study also focuses on the collection of functional assessments, such as the North Star Ambulatory Assessment, to assess the clinical effect of Suvodirsen, as the clinical benefit has yet to be established for any exon skipping approach. This will be critical information as we work toward more completely understanding the potential effect of Suvodirsen as a possible DMD treatment. The inclusion criteria and exclusion criteria, some of the key ones are shown here. Boys need to be between 5 and 12 years of age at the time of randomization. They need to be able to walk independently for 10 meters, 10 meters or more in 10 seconds or less. They have to have, of course, a genetically confirmed diagnosis with mutations amenable to exon 51 skipping. They have to have stable cardiac and lung function, and there are very specific criteria in the protocol that physicians, the investigators, will be able to measure that with. They need to have a stable dose of corticosteroids, any regimen, but they have to be started at least with 6 months prior to entering the study with no change in the dosing approach within 3 months before the trial. Again, the goal there is to try and get a stable baseline. No treatment. Previous patients cannot have had received treatment previously with Dystrophy, a gene therapy, or a PPMO. Participation is permitted for those previously treated with Adalurin or Eteplirsen after a 14-week washout period. As I mentioned, Distance 51 is designed to look at both dystrophin production as well as functional outcomes, including the North Star Ambulatory Assessment. In the U.S. and certain other regions, the primary endpoint is change from baseline dystrophin as measured by Western blot. In the EU and Japan and other regions, it's change from baseline in the North Star Ambulatory Assessment. The key secondary endpoint in each of these regions is essentially the reverse: the North Star Ambulatory Assessment in the U.S. and dystrophin expression in the EU and Japan. We will also be looking at several additional measures of function and strength, including stride velocity, using the Actimile wearable device, as well as assessments of quality of life. Again, the overarching goal here of this study is to have a complete understanding of Suvodirsen's effect as a potential treatment. Now, one of the innovative things here about the Distance 51 study was its acceptance into the FDA's Complex and Innovative Design Program, or its CID Pilot Program. The stated objectives of the program are shown here. First of all, to modernize clinical trial design, streamline and advance drug development, and inform regulatory decision-making. In order to get into this program, we had to submit an application that demonstrated our trial had innovative clinical trial design features and be in an area of high unmet need where there are limited or no treatment options. What we submitted was viewed as innovative for 2 reasons: by proposing innovative statistical methods to enable historical control data, meaning data from previous clinical trials and research studies, to augment the placebo group in our study. So why did we do this? And this is another area where we heard loud and clear the goal to minimize placebo exposure in clinical trials. And this was really one of the key elements behind us doing that. We heard loud and clear that there have been numerous trials and natural history studies this community has supported and continues to support that have enrolled boys exactly like the boys that we're seeking to enroll in our study. So we said, how can we use these data to support the efforts? And this is basically what we did. Through this approach, through this application, we met several times with our colleagues at FDA to refine the study analysis plan with a goal of reducing the number of patients required to deliver conclusive clinical efficacy results, potentially minimizing the number of placebo patients required for completion of the study, while accelerating study completion. And I have to say that as we have gone through this process, our colleagues in the overall DMD community, academic participants, other companies who have worked in this space, our colleagues at PTC, Pfizer, BioMarin, all have stepped up to try to get us these placebo data, to enable us to do this innovative approach to trial design, and we're quite hopeful that this becomes the standard by which future trials in this space are conducted. And it is our obligation as part of this program that all the details of how we're doing this become part of the communities, are disclosed publicly, and that is something that will be coming in the future. I mentioned we're currently recruiting. This slide lists the current clinical trial locations. Additional sites are continuing to be added and assessed. The best way to keep track of this is we are posting updates on clinicaltrials.gov. Many of you are familiar with this site. As sites are added, that's the place to go. And we're continuing to look at sites in all regions throughout the world. Now, I mentioned Distance 51 program overall was driven by our interactions with the DMD community. I mentioned some of those ways. This was not the only way the communities contributed to the Distance 51 design. We were asked to limit placebo exposure, as I mentioned. We did that. We were asked to minimize the number of biopsies, but still enable the possibility of an accelerated regulatory path. We've done that. That's why we have these 2 biopsies, but have enough data to be able to use to accelerate our regulatory path. We have provided and will continue to provide open label access to treatment for the boys who participated in the studies, again, for everybody, including those who were on placebo. We've continued to communicate and listen about our trials as we've continued the process. We've demonstrated our openness and our transparency. It began with our communication of phase 1 data shortly after completion of the study in scientific venues, and each step we've provided updates and open letters to the community. I mentioned we've committed to sharing the CID trial design elements and placebo data from Distance 51 with the community to enable future innovative clinical trial designs, and we will be continuing to work with PPMD and others on study awareness to ensure that those who may be eligible for the study can find a place to enter and that they have the support needed to ensure successful participation and study completion. Finally, one key part for WAVE is that this is just the beginning for us. Our Exon 53 program is on track to enter the clinic to enable clinical data readout next year. And we have numerous other exon skipping programs at various stages of preclinical development, all built upon the learnings from our Exon 51 program. So in summary, who are we? WAVE, we are a genetic medicines company that are utilizing this PRISM platform to deliver transformational therapies for Duchenne muscular dystrophy, as well as other genetically defined diseases. We have our phase 1 safety and tolerability studies are complete. We have an open label extension study ongoing where we will have data later this year. And we have the Distance 51 phase 2/3 study underway. We remain committed to the community with a lot of research underway to expand our exon skipping approach to 53 and additional exons. And finally, I just want to reiterate the thank you to everyone. It's been a really fulfilling process for those of us at WAVE, for me personally, as we have worked with everyone in partnership. Basically, everyone has been great: patients, families, advocacy groups, healthcare providers, our colleagues at the regulatory authorities. All been extraordinary, all working towards the same goal, and I look forward to continuing to work on this, Suvodirsen and other programs as we bring it and other therapies to patients who need them. That's it. Thank you.
Great. Thank you, Mike and Jeff. That was really wonderful. We're hoping especially for really good news. So a couple questions came in, and also I want to start with something. Just, this stereopure chemistry. Are there other applications of that where that sort of technology has been used in therapeutic development? Are you guys the first the first to do it?
Yeah. So, we are the first company to be able to figure out how to specifically develop stereopure oligonucleotides. So we're sort of leading the way in that. But one of the powers of this is that it's not limited to a certain approach. It's not limited to exon skipping. It's not limited to other oligonucleotide approaches. It's foundational for all different applications. So we have that has led to a very large pipeline at Wave, where we're looking at neurodegenerative diseases such as Huntington's disease and ALS and frontotemporal dementia. We're looking at other rare ataxias, like targeting a certain disease, spinocerebellar ataxia type 3. And we have multiple applications that we're working on with Takeda as one of our partners. We're looking at liver disease with our partners at Pfizer, and we also have some early data on ophthalmology programs. So we're the first one to be able to crack this nut, if you will, of how to do this type of technology. But because of that, the potential is enormous, which has led us into a variety of areas. And this observation early in the history of Wave, where we saw this greatly improved efficiency in muscle cells when targeting muscle cells, has led us down this path of hopefully having something different for DMD.
Yeah, that in vitro data was compelling. So and about everyone's really looking forward, I think, to those results from the open label portion of the phase 1. And you said the fourth quarter of this year. So are you, how much detail? You said you would have a community letter or present something at a meeting. How much detail will you give the community?
So
are you allowed to say?
Well, I think I can say in general terms. We have, when we have these data, we would be disclosing the amount of dystrophin that we see, as well as details about the safety profile. So it would have a full picture of type of data that we're seeing. I think that's what the community and that's what everybody's waiting for. So we would be disclosing it, not just in our obligations. We do have obligations as a small biotech to disclose, unfortunately, in a very public way to many stakeholders, but to the community itself, there would be disclosure, there would be communication, and then there would be hopefully presentation. Not hopefully, we would I hope to get into a scientific meeting to be able to share what we see. So it would be scientific venue, patient community communication, as well as our other obligations to our investment. Investors.
All right. Yeah, I think everyone really appreciates transparency, so that's wonderful. We had a question come in about sort of the applicability, right now it's for mutations amenable to exon 51 skipping. What about a 51 duplication? Would that work in your chemistry?
A 51 duplication. Yeah, I think that the applicability of the chemistry really can be for any type of variation you can think of. How we would evaluate that was something we'd have to think about. Technically, the stereochemistry allows us to design specific oligonucleotides as necessary to be able to target amenable, all sorts of amenable deletions and other ways that the gene is non-transcribed. We would have to think about whether a certain particular mutation, whether that would be, we'd have to figure out how to do it if it wasn't part of the normal, one of our existing programs. I think what you'll see is that our hope here is that when we have data from the Exon 51 program and our Exon 53 program, we will have a consistent approach that then can be applied to a variety of other deletions. But we won't know what's feasible until after we see our first data from Exon 51 and apply those learnings to other exons.
Okay, and do you have any plans to include non-ambulant boys at some point?
So we in our phase 1 study that was open to non-ambulant boys, we recruited patients up to age 18. I think we had about I think 20% of them were non-ambulatory, so we have a little bit of data there. But our commitment is to, once we understand in our existing studies the potential of exon skipping in these ambulant populations, we would expand into the non-ambulant populations. We're committed to doing that. In other populations not addressed by the study.
Okay. So I also want to just go back and just talk a little bit about the biopsy. So you, just to reiterate, so you said in the phase 2/3, everyone would go through, have 2 biopsies. And then I noticed on the slide, so these are going to be shoulder muscle biopsies. Can you talk a little bit about that?
Yes. Yeah. We had numerous meetings with patient groups as well as investigators about the appropriate place to obtain a biopsy, or what would be the best and most acceptable place. So we settled on taking a biopsy from the shoulder muscle, the deltoid, and that was felt to be the best place, given that the muscle of the deltoid at the ages that we were looking at would be of sufficient strength and quality to be able to assess the impact of exon skipping. Now, you could also go to other muscles of the upper extremity, such as the bicep or elsewhere, and we were strongly urged to go for the deltoid because the scar from an open biopsy would then remain under the short sleeve and of a boy's T-shirt, and we would, which took some pressure, we were told, off of the young boys from having to explain why they had a scar to their friends. So that's why we went focused on the deltoid.
Okay, and is there going to be, I would think the standardization and a central lab for reading all that, because I know in the past we've had some issues with that, so just want to get some clarity on that.
Yeah, that was probably one of the most robust discussions we've had throughout the entire process. Second to where the biopsy was coming from, how are you going to handle the biopsy so you don't screw this up? So we have had extensive conversations about how to do this, standardization both with the sites themselves as well as with the regulatory authorities. The regulatory authorities want very strict standardization, and the approach we are taking basically goes from, it's a standardized approach from the time the patient is brought to the operating room, and if it's done in an operating room and the muscle is extracted, right up to the point that it is finally analyzed, is regimented, and we have monitors along the way to essentially make sure those samples get to where they need to go, that they're the quality that we need, and that the analysis is performed appropriately. There are step checks every step of the way. The rigor is probably greater than anything I've seen. Again, this is our primary endpoint for the United States, so we've spent an enormous amount of time making sure it is done appropriately and making sure that this precious tissue that is taken is treated appropriately. This is precious. It's coming from under great dedication from the patient and their family. We don't want to be responsible for having a problem with that.
Okay, thank you. That's very thoughtful. So I have another question. So you said you're obviously exon 51 right now, and then exon 53 skip amenable. And so you were saying, how would you go after that? I mean, after that, you think, is it like sort of a platform approval approach, do you think, or with like just safety, or do you have to pick another exon? Like, could you elaborate a little bit about that?
Yeah, I would say that's something we would be working a lot with you on. I mean, you're very heavily involved in this whole platform protocol that PPMD is sponsoring. I mean, we have to figure out if there are ways to leverage all of the work that you and others are doing that enable us to facilitate a more rapid approach. I mean, the numbers get smaller and smaller and smaller after 51 and 53, and 45, as others know. And we are definitely aware that patients who happen to unfortunately have those more uncommon mutations have been left behind. So we're trying to figure out the most efficient ways to be able to do that.
Yeah, and I guess it's probably also a question of, how alike are those groups of patients with, mutations that are amenable to 53 or 51 skipping or 45. Are you going to get any of that information from your CID program from looking at those historical databases, or is that just for comparing placebo?
So we'll have access to the historical data set that we're going to have access to are placebo groups, but they'll be placebo groups not just restricted to exon 51 amenable or exon 53 amenable. They're data sets that include all different patient populations, which will basically be able to, working with other collaborators, to be able to hopefully, like CPAP, we're part of the DRISC initiative with Critical Path Institute, that we're hoping that some of the information from there helps us also facilitate development. I mean, they're helping us, as you know, quite a bit with our existing, with the Distance 51 study. So I think that's how we leverage those data sets to address these questions, and how we work with the regulatory authorities to understand under some of the newer legislation like cures whether we can somehow leverage our existing chemistry work. All of that remains to be seen, but we have to, it's not been done before, so we have to just figure out the best way.
But that also, I think this work also helps sort of define how you might be able to, we can bring these mutation-specific therapies into our platform trial. So, understanding the similarities and differences. Yeah.
Exactly.
These are all the questions that need to be answered.
We have to figure out how we can best leverage all of these ongoing initiatives.
Yeah. So actually circling back, so specifically when I asked the question earlier about, non-ambulatory inclusion, so we often define, trial participants, 18 up to 18. So is your definition, and maybe you haven't even thought about it yet, but is your definition of non-ambulatory going to be by age or by sort of the health status of the patient?
No, we have talked about that.
Okay.
Or based on health status. I mean, there's an artificial cutoff. We don't want to establish it. 5 to 18 was for the phase 1 study for a variety of reasons. Just because it was phase 1, we wanted as homogeneous a population as possible. When you're now expanding into different approaches for expansion into non-ambulatory studies, that's a different consideration. That based on the safety profile that we have to date, we can then leverage that information to understand how to go to a wider age range, and the restrictions simply are going to be around for safety purposes, for what age range we believe that fit criteria that we believe that the drug can be safely administered, not just 18. There's nothing magical about 18.
Okay. Good. That's the right answer. Yeah. No, I was just at another conference where it's like we're just arbitrarily making these cuts by ages. It's like, wait, that doesn't really make sense.
No, we're learning a lot. We're hearing a lot that there's so much variability in presentation here that that's not the way to be doing making these decisions. Right.
Well, and that's the same for other diseases too. So I have a question for Jeff. I think probably either of you, but probably Jeff. So I heard that you guys had a really great community, an input or advisory board, and that it had a really positive result. I think it was very beneficial for everybody. Are you going to continue to do those, or what's your vision for community involvement going forward?
You set me right up, Abby, without even us talking about that. But
okay.
No, thank you for that. And I mean, I would just reiterate what Mike said earlier, which is we would not be where we are without the input and support from the community. We have many different ways of getting that. Part of it is meeting with you, Abby, and the team at PPMD and the other organizations, getting your perspectives and your priorities. Meeting directly with the families at the conferences and community events out there. yeah, coordinating meetings with folks in the community. We actually we have one this weekend. To be honest with you, we have another one this weekend. So all of that information goes to all of our work, and so that gets incorporated into everything we do, such that everything we do again is to meet the needs of the folks that we're hoping to serve. And Mike showed you some examples of how we've actually, 4 examples of how we've changed our research. And so again, that is critically important. I put my contact information here. I always, am open, and I mean, this is my job to really understand your perspectives, your stories, and bring that into the company. So, thanks for pointing that out. And yes, we will certainly continue with getting feedback and collaboration with the community.
Okay, well, I don't have any more questions. So if there's any last things you want to say, Mike or Jeff, this is your chance.
Just to say thank you again. And, this has been, as I mentioned, quite a journey. And we know it's only the beginning of a journey as we enter into our phase 2/3 study. We are very much looking forward to, hopefully seeing some great data later this year and having that be the foundation of continuing to go to tackle this disease as best we can. We think we have a lot of potential to do so. And again, as I mentioned, this is the beginning for us, and we look forward to continuing to work together on this. So thank you again.
All right. Yep, I look and just make sure we have no last questions. I think we've answered them all. So really, I'm really looking forward to that data too. And you guys have just done a great job. Thank you so much for keeping us so informed and collaborating so well. So if there's no more questions, I think we will close the webinar.
Thanks so much. Thanks, Abby. Thanks to everyone on the line. Thanks, Kaylin. And look forward to seeing you soon.
Okay.
All right.
All right. Bye-bye.